Thanks, Shalini and Suhail for the invitation. I'm going to try to go through some very practical approaches for working with cancer genome sequencing data to produce copy number estimation. I'm going to start with a general overview with some quick intro slides, a little bit of background, a small discussion about read counting, and then discuss a couple of use cases, and then finally we'll get to the data with some with a demo. So I usually like to start talks with like this with an overview of what we can see, how we view a cancer genome. And until about 1993, this was our view, our high-resolution view even, of the cancer genome. 1993 started with CGH and Sky on chromosomes, FISH as well, and has progressed with the sequencing of the human genome. We've progressed to the point where we can actually measure cellular processes in the cell on a high throughput in a high throughput way for pretty much every step of the central dogma, starting with DNA replication right down through to protein translation, post-translational modification, etc. And although we're going to be focusing today on copy number, to view the phenotype, cancer in this case, and do it holistically, we really need to look through multiple lenses. We need to look at gene expression, transcriptional regulation, DNA methylation, sequence variation, chromatin modification, structural variation, as well as gene copy number. And it's really only by looking at all of those things, or as many of them as possible, and sort of correlating, asking questions about how one of these views affects another, can we really begin to understand cancer. So again, I'm going to focus on cancer here. And I know there are probably some people in the audience who are looking at copy number variation in germline, in the germline. I'm not really going to spend much time on that, and if there are questions about it, we can of course try to answer them. But really, again, I want to focus on cancer because that's what we're spending a lot of our time doing. And of course, copy number variation is very important in cancer. So some assertions that I'm making in moving forward. We're starting from the idea of using genome sequencing on next-gen sequencing platforms as the basis for this data analysis. And why would we do that? Well, because cancer is a genetic disease. Of course, we're interested in how base pair level changes affect or cause the disease that is cancer. And we can typically get that from exome or whole genome sequencing. Cancer is also a genomic disease, so it's not just a question of single nucleotide or very small changes that affect a single gene. The cancer genome is generally speaking disorganized. It shows copy number changes as well as structural variation. And then finally, an assumption here that we make when we're talking about cancer is that a comparison between a matched tumor and a normal genome is a useful approach for defining driver events in cancer. So as we move from a normal genome to from a normal cell through, in some cases, a premalignant cell, finally to a full-blown tumor, there are changes that happen in that genome, in the tumor genome, relative to the normal genome, and a lot of those are probably passenger mutations, passenger changes that are not necessarily driving the tumor, but of those changes that are different, that occur going from normal to tumor, some of them will be driver mutations, and that's the assertion or the assumption that we make when we're doing these comparisons. So why apply whole copy number to sequence data? Apply copy number analysis to sequence data? Well, it's because we can. And in a lot of situations, we don't necessarily want to spend extra money, for example, to produce a copy number. That's an obvious sort of trite way of thinking about it. So there are some others. You can use copy number data to quality control for sequencing results. In particular, copy number change is very common, and some might say ubiquitous event in cancer. So there are not very many cancer samples that we've seen that don't show some amount of copy number change. They could be actually simple copy number changes associated with whole chromosome gains or losses, or down to very disorganized genomes. And we'll see a couple examples of very disorganized genomes later in the talk. Copy number estimates. Copy number can provide a rough quantitation of normal admixture. And rough is a changing target, but the idea here is pretty simple. When we're looking for single nucleotide variants or small insertions or deletions in the tumor, particularly when we're getting samples taken out of the human body as chunks of tumor, those tumor cells are mixed in with normal cells as well. So if the amount of normal admixture is high, then any copy number changes that we can see are actually compressed toward looking more and more like normal. And why do we care about that? Well, if, for example, there is a single nucleotide variant that's present in 50% of the tumor cells, and we have on top of that a fair amount or even a large amount of normal admixture, we're going to be looking for essentially a needle in a very large haystack. And the copy number estimation can provide that a quantitation of how much normal admixture is there to let us know essentially how small the needle we're looking for actually is going to be. And finally, copy number profiles are somewhat distinct within tumor types and certainly within samples, and may be useful for finding sample identification, and I'll show an example of that later as well. Obviously, the most important reason that we want to use copy number analysis is to understand biology, and I think this goes unsaid. Copy number variation is one of the primary mechanisms for activating oncogenes and inactivating tumor suppressor genes, and copy number can provide biological insight regarding discovered SNVs and small insertions and deletions. So to give an example of the second point there, if you see a copy number, or I'm sorry, a single nucleotide variation that's expected to change protein function, one can ask the question, what kind of change in protein function are we going to see? Is it going to be an activating mutation or an inactivating mutation? And in cases where you see the copy number of the gene affected increased, that might give you a hint that the mutation is going to be an activating mutation. And that, of course, that copy number change can be in the sample in which you see the variation, or you can get that hint from other samples. So you can activate a gene by changing the protein structure, or you can overexpress it, which might have some similar results. The opposite is true for deletions. When you see deletions, you might see in a gene, you might think of loss of function mutation, or loss of function mutations. So some assumptions when we're talking about counting reads and estimating copy number. The density of the read counts in a sample will be proportional to the number of copies of DNA in the cell. So if we have a certain region of the genome and we count 1,000 reads in the normal sample, and we count in the same window 2,000 reads in the tumor sample, we might think, and we might assume that there are twice as many copies of the DNA in the tumor cell after accounting for the different read counts in the different samples. What follows from that is that comparing these read count densities between tumor and normal gives an estimate of the relative copy number in the tumor. And I'll just point out that it's a relative copy number, and you need to do some more steps to get to an absolute copy number. Some practical approaches that sort of fall into three large groups. We can count reads in fixed intervals across the genome in the tumor and the normal. For example, every 10 kb across the genome, we count the number of reads in the tumor, count the number of reads in the normal, and make a ratio, essentially, to come up with a copy number estimate, relative copy number estimate. We can count reads in fixed regions across the genome. For example, exome capture regions. So each exon in the whole human exome becomes a region in which we do counting, and again we make ratios. And we can also count reads based on windows of a certain number of reads. For example, windows of 1,000 reads in the normal sample. Use that to define sort of a sliding window, and then using the windows defined in the normal sample, go back to the tumor sample and count the number of reads in those windows. Again, coming up with relative copy numbers. The advantage of the bottom, or the last point there, is that we can take into account variation in read density, as, for example, happens in exome capture experiments, where we have very high coverage over the exome, but in the intergenic regions we have much lower coverage. We'd still like to use those intergenic regions, the reads in the intergenic regions, to help us estimate copy number. So what did the data actually look like? This is a screen capture from the Integrated Genomic Viewer from the Broad Institute. And what's showing across the top is chromosome 17. There's a small red bar that shows the region that we've zoomed into. So we're looking at an 85 kilobase region around the BRCA1 gene, which is noted across the bottom. The blue boxes are the exons, and the lines with small arrows are the introns. And you can see in the top panel, 01n.bam coverage and 01n.bam, and then the bottom panel, or the middle bottom panel, 01t sample. So a normal and a tumor sample. And the gray lines, or the gray area showing in the boxes, those are actually reads. And so where you see sort of waterfalls of gray, those are regions that were captured at high coverage, the exons, and in between we see very little coverage. If we move off of the exons, though, into a gene desert, we can see that there are actually reads in between the genes. You'll notice across the bottom there are no genes annotated, but we still have reads present. And we can use those non-captured reads, some call it the background reads, to estimate copy number between genes and improve our coverage uniformity. So a general workflow, and this is the kind of workflow that I think folks would want to think about if they wanted to do this themselves. You start with a normal sample, a tumor sample. You do the library preparation and sequencing, and that can be exome sequencing. It can be low-pass full genome sequencing, or a whole genome high-pass sequencing. We align the raw sequences to the genome, and the choice of genome is relatively important, but only to the point that we are able to compare from one analysis to another. You take those two output BAM files, that's the output of the alignment algorithm, and the input, those two normal and tumor BAM files become the input to the copy number estimation algorithm. After we do the copy number estimation, then we typically want to view the copy number estimates in some viewer, and in this case we're going to be using BioDiscovery Nexus. We like it because it's very fast. It allows a lot of downstream sort of processing, and is really sort of a one-stop shop for looking at copy number data. We do find that copy number data, unlike a lot of other kinds of genomic data, is a very graphical, requires a very graphical approach. It really requires someone sitting down and looking at the data. There's a lot that can be learned from applying sort of black box algorithms, but in many cases it's really useful and instructive to look at even individual samples to try to learn some biology. So copy number estimation and conversion to the Nexus format. So there's a postdoc in our lab and whose name is Josh Waterfall. He and I put together a very, very simple data package written in Python called ngCGH that can be installed fairly easily on Windows, Linux, and Mac. Usage is fairly simple. The output formats are described here, and after running this kind of analysis, we can convert from sort of a generic tab-delimited text format to something that can be used by BioDiscovery Nexus. And recently, we've gotten started to look at complete genomics data from tumor normal samples. And although complete genomics does most of the upstream processing and generates a quite useful file with a very long name, somatic CMV details deployed beta, we can convert that into a format that's understood by BioDiscovery Nexus as well. And we'll be seeing examples of that in a bit here. So the copy number estimation for Illumina exome sequencing is fairly simple. We've done the alignments to the human genome for the normal and the tumor. You would run a command something like ngCGH minus W 1000, use a window size of 1000 reads in the normal, then the normal BAM file name, the tumor BAM file name, and you get an output file. And the output file is very simple. It's chromosome, the start location of the chromosome, the end location of the chromosome, the number of reads in the normal, which is always going to be 1000 in this case, since that's what we asked for, and the number of reads in the tumor in the exact same window, so 854 in this case, which results in a log2 ratio just under 0. So a slight copy number loss. After we get the output from the copy number estimation, we can use convert to Nexus, part of the ngCGH package, to convert the output from the previous step into an output that's understood by BioDiscovery Nexus. In this case, we use the multi-1 format, and I'll show an example of how to get the data in a bit. But you can see that the data are actually quite similar to what we just saw in the previous slide, just reformatted. Some people, so the main parameter in ngCGH is the window size, and people might ask, well, what's the effect of using different window sizes? There's going to be a trade-off actually between the number of reads that we use and as I'll show here in a second, the noise that we get in the data. So a window size of 200 reads gives you a higher resolution, 5 times the resolution of the window size of 1000 reads. But as you can see, this is the same sample run twice with 200 reads and 1000 reads in the window. You get a much more robust picture of the data with using larger window sizes. And you can see that a little bit more specifically here. What I'm plotting here is the window size across the x-axis, 2 different samples, S14 and S93, that have different read numbers, And then DLRS is basically a measure of the noise. The higher the number, the higher the noise. And you can see that there's a nice relationship between the noise and the window size, whereas the window size increases, the noise decreases. And obviously we're trying to balance the resolution that we get with the noise that we get. So moving on to describe the data sets that we're going to be talking about. The exome capture data set is from 19 tumor normal pairs from melanoma data. This was data generated at NHGRI by Yardena Samuel's lab. It's been exome captured, mapped to the human genome, version HG18, and processed with a window size of 1,000 using ngCGH. This ends up giving an average of about 100,000 pseudoprobes per sample. It's more or less depending on the read depth that was used to generate that resulted from the exome capture. And we're going to also be looking at a complete genomics data set. The sequencing was done by complete genomics itself. A tumor and normal pairs. Data processing was done using version 2 of the complete genomics cancer pipeline. And then the CGI to Nexus command from the ngCGH package was used to produce the Nexus format. There were 10 tumor normal pairs from osteosarcoma, and this gives an average of about 1.4 million pseudoprobes, something like that. These are data from the Target Osteosarcoma project. And before we jump to the demo, I just wanted to go through some acknowledgements. From the Meltzer lab, Josh Waterfalls contributed to the ngCGH package. Paul Meltzer, Sven Bilke, Jack Xu, Yonghang Wang, Bob Walker, Marvin Pineda, Ogun Aban, Yuan Jiang, and Chris Lau have all contributed to the Target Osteo project and the sequencing associated with that. The Target Osteosarcoma Consortium has contributed the samples and a couple of SNP arrays that I'll be showing for comparison. The melanoma data was done with Yardena Samuels and Stephen Rosenberg, and then finally the folks at BioDiscovery Nexus who've been really supportive of our attempts to move these data into from the sequencing realm into the copy number realm. So at that point, I'll jump over to start looking at Nexus. I've created projects already, so I'm going to open an existing project. And the first one that we're going to be looking at is the Melanoma Exomes. Just a note, this is being run on my Mac laptop, so this is not a supercomputer. It's quite simple to use these things and pretty fast too. So when you open the data, a data set, what you see are the samples listed here, the status, whether the data's been processed or not. The data type in all cases you'll see is multi-1, and what that means actually is that the file format that we used is specific for Nexus. But it also, there's also an interesting point here. Every probe set from each of these different samples has a different location. So the probes from the first sample here don't match the probes from the second sample, don't match the probes from the third sample. And BioDiscovery Nexus is fine with that. And in fact, as you'll see later, we can actually mix and match even different platforms. So let me just show you how I actually loaded the data with one sample. The sample name is 231000 CGH Nexus, and I chose the type multi-1. There's no probe type here, so I've now loaded the data in, and we can view the data. And just to give an example of what the data actually look like, we can well, first an overview of the data here. Across the top, we see the different chromosomes. Blue is amplification or increase or chromosomal gain, and the dark blue is high-level amplification. Red is loss, and the darker red is basically homozygous loss. And you can see that there's a nice gain of chromosome 7, which is very characteristic of melanoma. So let's look at what the data look like for a single sample. So I'm just going to choose one sample here randomly, and we can see some information about some general information about the copy number gains and losses. And if we click on the whole genome view, we can see now across the whole genome what the data look like for this particular sample at the level of individual probes. The black lines represent the copy number estimation by segmentation, and you can see that this genome is broken up into many pieces. And you can also get a sense of the spread or the noise in the data as well here. So that's a single sample. I loaded another sample. This was the sample that I loaded separately. And we can also look at it. And if we look at this sample, we can see that although we can see copy number changes and sort of general trends in the data, these data are very noisy. And this doesn't happen too often, but it does happen. So so just because we have the good sequencing data, we don't always necessarily produce comparable tumor and normal sequencing coverage, so it can become difficult to estimate copy number. So in this case, I'm going to exclude this sample. So we're going to go back and just delete it. So now we can look at what the data look like. Now we can begin to do some more interesting biology. So I'm going to search for. I'm going to search in the search box for P16 CDKN2A. Click Find, and you can see that happened pretty quickly. And what we're seeing across the top again is for the whole sample set, we see that actually 63% of the samples show a loss, and nobody shows a gain. P16 is a known tumor suppressor, so we would maybe expect this. And I should have mentioned these are actually cell lines, so it wouldn't be unusual to see a P16 loss in cell lines. By clicking here, I can sort the samples based on essentially copy number. And if I click on a single sample, I can see what the copy number profile looks like for that sample. And if folks have looked at copy number data before, you'll recognize that a copy number estimation with a log 2 ratio of -9 is pathognomonic, or it means that this CDKN2A region is gone. There are essentially no reads in this location at all. So a total deletion. Here's another sample that shows a similar effect. We can do the same thing for P53, for example. Here's the region. It does show a couple of samples that show some slight gain. And again, clicking on the region, you can see that this region is just non-existent. The copy number is 0. So we can play that game for a long time, looking through individual genes, but Nexus allows us to do some other things as well. So we can use a tool called Gistic, which is essentially a way of looking at recurrent copy number changes in a statistical framework to define the regions that look like they're most likely to be driver copy number changes. I'd refer you to the paper if you're interested in the details. What I just did was to sort the resulting Gistic output by G-score, which is a measure of how strong the Gistic results actually are. And by clicking on the regions now, the Gistic pulled out as being important drivers, we can get the names of the genes. And the first region, the most statistically significant region, contains the PTEN gene. And or for those of you who are cancer geneticists, you recognize that as a very likely candidate for being a tumor suppressor and showing loss. So let's pick the next region here. If we look in the extended region, and I am doing a little bit of cherry picking, but you can see how quickly you can go through these yourself. And in this region, we see CDKN2A. So the second highest hit from Gistic includes another tumor suppressor. And if we go down the list a little bit further, down to chromosome 17, we can see in this region P53. So in the top 7 or 8 regions using Gistic, we've picked up 3 known tumor suppressor genes that look to be statistically interesting from the point of view of recurrent loss. So again, with Nexus or with tools like Nexus, you can spend a lot of time playing with your data, and you can see how applying some of these different tools, you can get biologically important information fairly quickly. So I'm going to move on now to the second project, the Osteo complete genomics data. And you'll see I have actually 10 samples labeled sample 1 to 10, but I also have a couple of samples, sample 1.AFI, sample 2.AFI. I'm going to remove the AFI samples from our view for the time being, just so we can see what the other data look like from complete genomics. Now remember, these are now complete genomes rather than coming from exome data. But in Nexus, it all looks the same at the level of copy number. So let's view these samples. And you can see, again, across the top, the copy number profile for osteosarcoma. And just to get a little bit of a sense of how these data look different than the melanoma samples, I'm just going to pick a single sample here, sample 3, randomly. This has many more probes, so it takes a little bit longer to load. What we're going to do is just look at sample 3 in some detail. If we click on the whole genome view, you can see that this is a pretty disorganized genome with lots of high copy number gain, lots of loss, and in fact it's actually hard to pick out what's normal. And just to give a better view of what the data look like at a fairly high resolution, I'm just going to pick chromosome 4 because it's quite telling of how osteosarcoma often looks. This is unfortunately for the patient a typical osteosarcoma genome, and you can see lots of gains, lots of losses, and actually very little that you would be able to pin your finger on as being normal copy number. Again, this is relative copy number, so we can't really say quickly and easily what the actual normal copy number is. There are some ways to try to get at that, and some algorithms for doing that, but I haven't applied those here just for simplicity's sake. again, we can do the usual search for a gene of interest. I'd like CDKN2A today. Again, our friendly CDKN2A location. Another example of chromosome loss associated with this associated with that gene. And again, you can play that game for a long time. But I wanted to show with this data set some more general aspects of looking at copy number. So I'm going to add in the AFI samples. Oops. Add in the AFI samples, and then go back to the data set view. And Nexus has this nice little tool called cluster. And what it's done is to, in the same way that, in a similar way anyway, to clustering gene expression profiles, Nexus clusters the copy number profiles. And you'll see why this is useful, particularly useful in a bit. Even though these samples were done on two very different platforms, the AFI platform and complete genomics processed through to copy number, the samples nicely cluster together. And this, of course, is not a given. So if you had a situation where you had multiple estimates of copy number, you can use this kind of simple analysis, simple visualization tool to get at some sample identification, sample identity issues. Let's go back to the dataset view, and I want to isolate now down to the samples that are run on the AFI and on the complete genomics platforms. I'm going to sort the samples by sample name so that sample 1 is next to sample 1 AFI and sample 2 is next to sample 2 AFI. That's as simple as clicking the button here. Click View. And now what we have are two different copy number estimates for the same sample. And you can probably get a little bit of an idea that the samples actually do, I should zoom in a bit. Clicking the bottom bar here allows us to zoom vertically. And you can get a sense that the two samples actually do agree at least in general with each other when you're comparing the AFI and the complete genomics data. We can zoom in a little bit further, though, and look at the individual probes and get a sense of what the operating characteristics are of sequencing data and AFI SNP arrays. So in this top panel, we have sample 1 estimate, copy number estimates from complete genomics data. In the second panel from AFI, sample 2 from complete genomics data, and sample 2 from AFI. And you can see that there's some quite distinct similarities between the two samples. Copy number gain here, normal copy number loss here, loss here. But you can also see that there are some significant differences, particularly in the noise profiles of the two data sets. And I'm not trying to make a claim that one is better than the other. There are some details here that come in as to how the data are processed and the qualities of the individual arrays or sequencing reactions. But you get a sense that at least the copy number data are at least as good as the arrays in this case anyway. Going back to the dataset view, I haven't done things with the sample annotations, but if we did have sample annotations, then we could add those sample annotations as factors, and we can then do comparisons in copy number profiles between, say, for example, stage 1 and stage 2 cancer, just as a simple example. And since we have, since we're talking about sequencing data, one of the factors that we could add is actually single nucleotide variation. So as an example, there are a couple of ways to, using the P16 CDKN2A example, there are a couple of ways to inactivate P16. The most likely or the most common, is deletion, but you could actually introduce a mutation that's inactivating, or a small insertion or deletion that doesn't necessarily show up as a copy number change. And if you had the samples annotated using that kind of information, you might be able to see that, for just as a hypothesis, you could test that samples that show an inactivating single nucleotide variation or insertion deletion event are not, don't show homozygous deletion. And those that show homozygous deletion, of course, we wouldn't actually have sequencing data for. The data just wouldn't exist. So you can begin to draw those kinds of correlations. I think at that point I'm going to stop and entertain any questions. For the Nexus side of things, the Nexus folks have much more experience with working with the software itself than I do, so questions along those lines we can direct back to them as well.
Thank you, Dr. Davis. That was a very nice presentation. We do have several questions. I'm not sure if we'll have time to go through them all, but we can attempt to do so. One of the questions, I think it's referring to early on in your presentation, is you mentioned admixtures. Are there methods to analyze copy number variation in admixed populations, such as the Hispanic population?
So copy number, admixture here I'm talking about is admixture of tumor and normal, or if folks have seen recent New England Journal of Medicine paper, tumor from one region of the tumor to tumor in another region of the tumor being mixed together. That's the kind of admixture I'm talking about, and what that tends to do, if you think about what copy number estimation should result in, if we have a diploid genome and we lose one copy, we should see a decrease from a log ratio of 2 to a log ratio of -1. And if we don't actually see that, if we see that the mode for regions that show loss is something less than -1, then that might be evidence that that there's normal admixture, so that we're seeing a mixture of one copy loss on top of a diploid genome. And the extent to which that copy number estimation is much less than 1, that's evidence for more heterogeneity, more normal admixture. In terms of population admixture, which is I think where the question of Hispanic the Hispanic population comes in, there are copy number variants in the germline, which are very likely more strongly associated with one population compared to another. And in thinking about germline genomics and genetics, you'd need to take that into account when looking at things like association of copy number changes in the germline with disease. In this case, we're always comparing, or we're trying to always compare to an associated matched normal when we're looking at tumors. So copy number variation that's in the germline that also shows up in the tumor looks like a relative copy number of 1, so no change. That's an advantage in a lot of ways, but it's a disadvantage in others.
Okay. Thanks, Dr. Davis. Another question here. How much of a concern is it that you have unequal or biased hybridization based enrichment for these regions, which is producing artifactual copy number variants using a read depth approach?
It's a real concern. There are so one thing that I haven't typically done, but that BioDiscovery does for known platforms, is to correct for global GC content, which is known to affect hybridization. And in terms of local artifacts, what we rely on when we're looking at copy number data is estimation of copy number that doesn't rely on a single probe or a single genomic locus, but multiple genomic loci simultaneously, the so-called segmentation problem. And we hope that accounts for very small local hybridization biases. But I think, when I loaded that that one sample that showed quite a bit of noise, I think that one possibility is that the hybridization reaction was handled differently in one sample in the tumor versus the normal. Another probably more likely explanation is that the data in either the DNA in either the tumor or the normal was of lower quality. So some regions of the genome were represented better than others, leading to variation in copy number. In practice, I'm not saying that that this is a foolproof method, and as with all copy number estimation, you have to take it in a sort of biological context. So single changes, if you don't have other biological information or other information to make that important, you might have to move on, I guess. It's a non-answer to a question, but having something like Nexus to be able to look at the data is really a key first step to being able to answer the question of how well the copy number estimation actually worked.
Okay, thank you. Another question, have you done any exome CNV work on DNA extracted from formalin-fixed slides?
I'm trying to think if we've actually formally analyzed them. We have done those things. I don't have off the top of my head what the noise characteristics look like. In general, we found that formalin-fixed samples work reasonably well for exome capture and for other small targeted capture reactions. But unfortunately, I don't have a good answer, a good quantitative answer.
Okay. When you create your BAM files, what mapper do you use, and do you realign around indels? Do you remove duplicates?
Yeah, so the context of the question is, does it make a difference? And my guess is it doesn't make a difference what alignment algorithms you use in terms of copy number analysis. In terms of the steps after the alignment algorithm is applied, realignment around indels and marking or removing duplicates. Of those two, probably the most important one is to remove duplicates. We do have samples that show a high degree of duplication, probably due to low complexity DNA going in, so low quality DNA going in into the reaction. But that's the one of the steps. That's the step that's most likely to help you clean up the copy number estimation. To answer the question directly about what mapper we use, we don't have a quote-unquote standard. we use BWA, Novo-Align, GSNAP, Bowtie 2, and ELAND, all of those in the lab.
And is there any way to normalize sequencing data between samples that were sequenced on two different machines?
In terms of normalizing, I'm not sure which question we're asking, but if we're talking about between tumor and normal done on different machines, we've never tried to do that. My guess is that might be a bit challenging. But if the tumor and normal samples were done on the same machine and then repeated on a different machine, I think that you can get a sense from the data that I showed here, even between the SNP array and complete genomics, that the results are actually pretty reproducible. And that's certainly been the case when we've looked at the exome data compared to the complete genomics data for the osteosarcoma samples, for example. That the technology itself is not so important. The most important thing is a match in the processing of the tumor and the normal samples.
Thank you. Another question here is, can this be applied to normal and control samples in non-cancer genetic diseases?
I think it probably, it probably can, but we haven't done it much. We haven't actually done it at all. And the choice of controls will be important. There's another piece that goes along with this. In some contexts, there may be better ways at getting copy number. Let's say more precise ways of getting copy number than using strictly read count based methods. So using the properties of paired end data on whole genome sequencing, you can use the read count information along with the paired end read orientations and distances from each other to potentially get a better estimate of small SNVs, which are probably more likely to be associated with disease on a population level than in this case in cancer, where we're talking about fairly large regions of aneuploidy.
Great. And for cancer genomes, how much depth of coverage do you need? Is it different from normal non-tumor tissue sample?
So the coverage needs are different for copy number than for variant calling, and typically the copy number, the coverage needs for copy number are relatively low. There's a paper from Eric Lander, I believe, in 2009. I think it was in Nature Methods, comparing SNP array resolution. I think it was an API-SNP6 array resolution to about 14 million reads from, shotgun sequencing basically of a sample, and the two were nearly equivalent. So a very small amount of sequencing data these days, a very small amount of sequencing data can give you something very similar in terms of copy number to arrays. Typically, the coverage requirements are based on the ability to call variants, and that's plenty high enough to do a reasonably good copy number estimation.
Thank you, Dr. Davis. Unfortunately, we're running out of time, so at this point, I'd like to take a little break and run the poll questions. And there are several other questions. Maybe we can cover one or two more after the polls. So I would like to launch the polls now. So it'll just take less than 2 minutes. Here's the first question. Here's the second question. Here's the third question. And here is the last question. Okay, that was the last question. Thank you everyone for participating in that. Unfortunately, we are out of time and I don't want to keep anyone, including the presenter, from other obligations. But we do have other questions that we can follow up via email. Again, I would like to thank Dr. Davis for the wonderful presentation. And if you have any further questions, feel free to email us at info@biodiscovery.com. And just another reminder, once the browser window closes, another one will open up where you can enter in any additional comments or questions you may have or give us any feedback. So feel free to use that for any additional questions you have as well. Again, thank you everyone for participating and thank you so much, Dr. Davis. Have a good day, everyone. Bye-bye.